All right, today we're going to be talking about how solutes and water are reabsorbed back into the blood from the kidney. So solutes are filtered in the kidney, and they're actually filtered at the nephron. And then, so this is a cortical, well, it's a nephron, and we mostly have cortical nephrons. And usually they don't look like this. I've just drawn it out this way to have it make more sense as I'm teaching. But this is the glomerulus. This top part, it has a capillary bed, and that's where things are filtered. And then we have the PCT, or proximal convoluted tubule, the PST, proximal straight tubule. It's a bad S, it's all right. And then we have our loop of Henle right here, and it's the descending tubule and then ascending tubule. And then as we go up, ascend, we have some macula densa cells, which we'll talk about. They're really important. And then we'll have the collecting duct, the distal tubule, or I'll just write collecting duct. And this isn't perfect, but this is a good drawing to help us understand what goes on where. And then we have a distal straight tubule somewhere. Distal straight tubule. Okay. And then in each of these points, they reabsorb different stuff, whether that's water or different solutes. So I'm going to draw a separate cell to go through the pathways of each. So each of these spots have different cells that do different things. So, okay. So I've drawn a cell in the proximal convoluted tubule, and we're gonna draw what solutes go in and out of here. And so this cell though, this would be the lumen side, or apical, and this would be in the blood, or the basal side of the cell. So we have stuff filtered is in the lumen. The PCT filters quite a bit. It filters glucose, amino acids, lactic acid, water, calcium, and potassium. And so we're gonna go through how each of these things go through. So we're gonna start with glucose. So there's a bunch of sodium in the lumen. Pretty important. All cells have a sodium-potassium pump, right? 3 sodium to potassium pump. It's the most important cell or pump in the cells. Just helps keep gradients and keep things moving properly. But for glucose, we'll have sodium come in, and it's a symport, and it'll carry in glucose. Bad handwriting, that's okay. And this gate is actually called SGLT. Okay, so that's glucose. Then we have amino acids, and sodium will also bring in amino acids. Let's try AAs. We have another one, sodium will bring in lactic acid, so LA. And then we also have aquaporins. We'll just draw this down here. Aquaporins, water will come in and water will come out paracellularly. Or no, sorry, this is transcellularly, also paracellularly. If we were to draw another cell, we'll just do that. In between these gaps, water can go through. That's pericellularly. And what's cool about this is, so with our pump, we've set up a gradient of a lot of sodium, so water will follow sodium. But there's something called solvent drag, and this water will actually drag calcium and potassium in with it. So that's another way we get calcium and potassium reabsorbed back into the blood. Okay, so what am I missing? bicarb. So we have a cool little thing here. To get bicarb reabsorbed, oh, real quick before I go into that, all of these will have them on the basal side as well. So glucose will go out with sodium, kind of the same thing. Sodium will pull these out. And then water will follow as well. Water follows solutes. And so we actually have, we'll stay at 300 milliosmoles of concentration of solutes as all this flows out. But bicarb, so we have a bunch of H's in the cell. Sodium will come in, sodium does a whole lot, and we'll drive H's out. And we have bicarb out here. Oops, HO3 minus. So the H's will flow out and join with bicarb, and through, I don't know what you call it, it just changes. But there's an enzyme to help catalyze this reaction. The reaction happens, and this enzyme is carbonic anhydrase. Okay, so sodium will drive out an H, which will react with bicarb through this enzyme, so it happens a lot and quickly, and it will turn into CO2 and water. The CO2 will actually diffuse across the cell. It doesn't need a channel, it's just so small and it fits the right criteria that it can just diffuse across the cell. So now we have CO2 in our cell. It will also react again with carbonic anhydrase, and it will react with water to then split off. We have H pluses again, or hydroniums, as well as bicarb. So this is pretty sloppy now, I'm sorry, but H pluses come out, get with bicarb, CO2, make CO2, CO2 diffuse across the cell, reacts with water, makes bicarb. And then we have a bicarb channel, and sodium actually, I'm gonna draw it out here. Sodium will drive bicarb out into the blood. So that's how we get bicarb from the lumen into the cell and into the blood. Another thing I forgot to mention, this glucose. This gate was SGLT. This glucose one on the basal side, or facing the blood, is called GLUT2. Okay, so that's the proximal convoluted tubule and the solutes that transport and how they're transported. Okay, now we're going to talk about the proximal straight tubule. So, use green this time for fun. So the main point of the proximal straight tubule is to get chloride into the blood. So we filter chloride, we have a bunch of chloride out here, and we have a channel where we have an anion here in the cell that will come out and drive chloride back in. I'm actually drawing this side. So the anion comes out, chloride comes into the cell. And then we have a leak channel where chloride will just flow out of. This is called CFTR, this leak, the chloride leak channel. And so, but we have a little bit of a problem. We need more anions back in here. So there's another channel where sodium will come in, good old sodium, and drive an H that we have in the cell, a proton, an H plus, back in the lumen. And our anion is also back into the lumen. And somehow these will combine to form, so anion plus H, they'll form a molecule that can diffuse across the cell. And then they'll split back to their H pluses and anion. That is terrible. That's okay. So that's how we get anions back in here to drive chloride in. And then, like I said, chloride will just leak into the blood. Okay, that's the proximal straight tubule. Okay, now we're going to talk about the descending tubule. And it's pretty simple. We just, we're talking about water. So this is where a bunch of water is going to leave the lumen and go into the blood. So we just have aquaporins here. So H2O is going to come in and just come right back out. Like I said, I failed to mention on the previous cell, but we also have our NAK pump. It's in every cell to create a gradient for water to follow. Water will also continue to go paracellularly, like we talked about previously. And so up to this point though, we have not changed the osmolarity. This is where it starts to change. So we had a 300 osmolarity. It's gonna start to drive it down. And eventually, down at the bottom at the loop of Henle, we're gonna see 600 milliosmoles. Because more water is leaving without solutes, so we're gonna have a higher concentration. And so that's the main point of the descending tubule. All right, now we're gonna talk about the ascending tubule. And this, at this point in the nephron, if you remember our little nephron, yeah, you can see that. We're on this stretch now, we're going up. And at this point, we're gonna stop becoming permeable to water. So we won't have aquaporins, and we also won't have these gaps. So we had a cell, right? Whatever. And we have these gaps that water could flow through. Well, these are gonna come to, well, I don't know if they fuse, I don't really know what goes on, but basically water cannot get through. Just bounce off. So also you notice, as we went down, our osmolarity went down to 600. We're gonna see as we go back up, the ascending, that 600, is gonna rise again. Because in our ascending, we're gonna transport a bunch of solutes. And so it'll drive that osmolarity back, I guess, back down to 300. So kind of cool. But we'll have a channel here. It's purple. And it's a symporter, and it's going to carry 3 things in: sodium, potassium, and 2 chlorides. And so all these things will come in from the lumen, and we're going to get them out in the blood. Remember, we have our NAK pump, so it's going to get the sodium outside. We also have leak channels. We have a potassium leak channel, and then we also still have that CFTR, as mentioned previously, where chloride can come and leak out. So we're going to get a whole bunch of these solutes from the lumen into the cell and into the blood. Let's see. Tight junctions, that's what these are called. I don't know why I wrote that, because you can't even read it anyway. They're tight junctions. And then one thing that's cool, we can actually block this gate, this channel, with diuretics. And so less solutes will get into the blood, and so we're gonna pee more, which is cool. If we need to, we can use that to block these channels. Anyway, so that's the ascending tubule. Okay, at the back at the top of this nephron, we have our macula densa cells, and we're still not permeable to water. But we're gonna move some more solutes from the lumen to the blood. Purple again. This time it's just gonna be sodium and chloride. So we're still moving a bunch of chloride. Let's see. And that's important. So we still have our NAK pump here. But we also have this channel that chloride will go out and bring in bicarb, HCO3. So we're gonna get bicarb back into these cells. That's pretty simple. That's the macula densa cells. Oh, so if we have a bunch of NaCl, they'll come in and these cells will sense it and will release adenosine or nitric oxide, depending on what we need and what we want. And so we're gonna go into that regulation a little later, but that's what the macula densa cells do, is it senses this NaCl, if there's too much or too little. All right, almost done. So now we're on our distal or collecting duct. Part of the nephron. And so there's a bunch of stuff that goes on with this. Let's use green and blue. So we have a channel. It's called the ENaC channel, and sodium is going to come into the cell. And then we have our NAK pump, right? Like always. We have actually aquaporins on the basal side of the cell, but we do not have them yet on this side. And so we can't get water through. And at this point, we want water to get through. But we have this vesicle in here with aquaporins. And we want this vesicle to bind with this apical side and form aquaporins. That way we can get water through. So we have this G-coupled protein receptor. G-protein coupled receptor. It's actually an ADH receptor that we have on here. ADR, I don't know why I wrote that. So when we need to get a lot of blood out, we send out ADH. ADH will bind. This will basically make this vesicle fuse, and so now we form aquaporins on this side. So water is going to come in, right, H2O, and it's going to come back out with this other aquaporin. So release of ADH will make water go from the lumen and reabsorbed back into the blood. We also have leak channels. Change back to green. Potassium leak channels. So they can go where they needed to be. Yeah. Oh, so a big thing, these are principal cells. So collecting duct, we have 3 different types of cells, and these are the principal cells. Okay. And then next we're gonna talk about type A and type B cells in this collecting duct. Okay, now we're gonna talk about intercalated cells in the distal duct or collecting duct. So type A cells, they do work when we're acidic. So if we have too much H's in the blood, what's cool is we have these channels where potassium will come out and bring an H in. Draw little pluses. Because we have too many H's out here. Then we also have one on this side that will push the H's out and bring potassium back in. So that's when we're too acidic. I'm just gonna, actually I could have just erased the letters. Now if we have a type B intercalated cell, these work when we're too alkalytic. So we don't have a lot of H's out here. We have a ton of bicarb. And so we want H's out here to balance it out. So now it'll bring in H pluses, and potassium will leave. And then we have another one that will push out the H pluses into the blood and bring in potassium. Whoops. So that's pretty simple. That's how the type A and type B intercalated cells work. One last thing that I forgot to mention in our PCT. Specifically, it's a problem when there's glucose. I'll draw a different color. So our glucose transporters are protein transporters, and there's only so many, so they can get saturated. So when there's too high of glucose, not any more can be reabsorbed and pushed out into the blood. And to the point where it cannot reabsorb any more glucose is our renal threshold. I'm just gonna write in here. Renal threshold. And this is bad. If you just have really high blood sugar and you have way too much glucose in there, it will not get reabsorbed and back in the blood. But it will stay into the lumen or the tubules, and glucose, yeah. So if it's not reabsorbed, we're gonna urinate out glucose because these protein transporters are saturated. And that can happen with lactic acid and amino acids, but this is a big problem with glucose. Okay, that's how we transport solutes in water. All right, now I'm gonna explain how filtration happens in the renal corpuscle. So this is the glomerulus, and we're first gonna talk about a couple pressures that drive filtration. So we have a hydrostatic pressure of 55. And that's kind of the same thing as in our blood when we talked about capillaries, is we have blood flowing through here, and that pressure is at 55. And then we have albumin in the blood, just like when we talked about in the past unit. So we have a BCOP of 28. And this is measured in millimeters of mercury again. And so that will so the hydrostatic pressure will drive water out. I'm gonna just write out. And the BCOP will drive water in, because water is gonna be attracted to the albumin in there. And so it's gonna drive water in. We also have a corpuscle, or let's see, maybe capsule pressure is what we call it. But yeah, capsule pressure, which is 10 millimeters of mercury. And it's also gonna drive water in. It's just like the pressure that remains here, it's just gonna drive water back into the blood. So we have a net pressure of 17, whoops, sheesh. Reset to around 17 millimeters of mercury. Okay. How we filter is through these capillaries and membranes here that we're going to talk about. So as blood comes in carrying a bunch of solutes that we need to filter through the kidney, we have these fenestrated capillaries. And so there's just holes in the capillaries where things can pass through. And then we have this basement membrane here I drew in purple. And this has a negative charge. So anything with a negative charge is going to be repelled. So some criteria we can use so far has to be no charge for something to be filtered through. It also can fit through, let's see, it is about 7 millimeters, not millimeters, let's see, size. So it has to be small as well. Nanometers, that's how much of a size it can fit through. And then we also have these podocytes, which I drew in green here. And they also are negative charge. And the size is like 4 to 6 nanometers. So it's even smaller. So it's a really good filter system. And so we need something small and no charge to be able to fit through. The blue I drew is just mesangial cells. They just help with filtration. We can, here, let's see real quick. Yeah, so we can regulate how much blood and the pressure that comes into this glomerulus. Change markers. So this side we have an afferent. Arterial. I-R. Sheesh. Handwriting's terrible. And this is an efferent. Arterial. And so if we have a low blood pressure and we want to increase the blood pressure, we actually dilate this. And when we dilate or relax, it actually lets more blood in, and so it creates a higher pressure in here. And then if we have what's called a tubuloglomerular feedback, I think it's what it's called, and we'll go into that later. But these smooth muscle cells actually can sense high blood pressure. And so when it senses high blood pressure, they react and they constrict. And when they constrict, we make this hole smaller. And so less blood can come in, creating less of a pressure. So it's mainly in the afferent. The efferent can do that as well, constrict and dilate to raise or lower blood pressure, but this is the most important. But not to be confused with when we talked about blood vessels in the past unit. If we're constricting, we're lowering blood pressure in here. If we're dilating, we're raising the blood pressure in here. Okay, a little more on GFR. GFR is important because it stands for glomerular filtration rate, and we can see how effective the kidneys are at filtering stuff through the kidney. So GFR. And our kidney compensates for this. If we have a low GFR, it activates a bunch of stuff to raise that up. Or if it has a high GFR, it does the opposite. It wants to lower it. So it maintains homeostasis. And if we come into the doctor and have something wrong with our kidneys, they can test for GFR. And how they do that is a concept called clearance. So GFR is how, the rate at which the glomerulus filters stuff through the kidney. But clearance is measuring a specific substance, how fast a specific substance can be filtered and cleared out through the kidney and urine per minute. We can measure it per minute. So there's an equation to help us. We'll put equals. So clearance equals the concentration of the substance in the urine and multiply it by V, which equals the flow rate of urine, and divide it by that concentration of that substance in the plasma. And that will give us our clearance, how much of that stuff is passed through the kidneys. And the reason this is important because this will help us measure GFR. So we need and this is just a specific substance. So if we find a substance that can match GFR, then when we test, we can just test for that substance, plug it into this equation, and we know if our GFR is low or high. Some criteria though, for our substance, to be able to match GFR normally, in normal conditions, it has to be small and uncharged, And then it cannot be, so that way it can filter freely. It cannot be, whoops, reabsorbed or secreted. So whatever filters through, it's gonna just keep passing through and pass to the urine. And it cannot be metabolized or made by the nephron. So we found a substance that fits these criteria, and it's inulin. And inulin passes through, it's small, uncharged, fits that criteria, and it filters through at the same rate as our glomerular filtration rate. And so as it passes through, well, okay, so inulin, we'd have to put in the inulin and then put the concentrations we find through that equation, and we can figure out GFR. But then we found a substance that's made in the body called creatinine, or creatinine. I don't know how to say it. But it's about the same as inulin. It's very small, it's uncharged, it can filter freely, it is not reabsorbed or secreted, and will pass through. And so we can take a measurement of creatinine in our urine and in our plasma and get our GFR, which is pretty cool. Okay, now we're gonna talk about some feedback systems to keep our GFR in normal range. So we have the tubuloglomerular feedback, and that happens in the macula densa cells. We've already talked about those. But in their macula densa cells, we sense concentrations of NaCl. So if we have a high NaCl, the macula densa will take this and release, why am I drawing a blank? Adenosine, sorry about that. Adenosine. And adenosine, what adenosine does, it vasoconstricts. Basically, if we have high NaCl, it means we have a higher blood pressure. And remember, in our glomerulus, if it's a high blood pressure, we want to lower it, and so we vasoconstrict to lower it because less blood gets in it. So that will lower blood pressure and also lower GFR. Okay, if we have a low NaCl, then we're going to release nitric oxide. And nitric oxide, you can barely still see that, yeah, will vasodilate. And then it'll raise blood pressure because when it dilates, it lets more blood in to the glomerulus, as well as raise GF. Oh, okay, kind of sloppy. But that's the tubuloglomerular feedback. So now we're going to introduce the Ross system. And it's another way of regulating GFR. So if we have a low GFR, we're going to release renin. And renin will turn into angiotensinogen. Yeah, you can see that. And then it'll turn into Ang1, angiotensin 1, and through an enzyme called ACE, we convert it to Ang2. And Ang2 can do a bunch of different things. Very cool. It can send a signal to the brain to increase thirst and increase ADH, or release ADH, antidiuretic hormone. So we're gonna wanna hold on to the water, and then thirst, we'll drink water, so it increases blood volume, which will increase blood pressure. We'll vasoconstrict. Ang2 will. And this is talking about the blood vessels, so we're gonna increase pressure everywhere. We're not talking about just the afferent arterial. It will vasoconstrict the efferent arterial, which would increase the blood pressure in the glomerulus. And then it also release aldosterone. Let me make sure I have that right. Yeah, it'll go to the adrenal gland and release aldosterone. Okay, and aldosterone, so we're gonna talk about ADH and aldosterone the most. Let's slide this over. So if we have a cell, black marker, there we go. What aldosterone does is, remember we have these ENaC gates? And sodium comes soon. We have the sodium potassium NAK pumps, which gets sodium out into the blood. Aldosterone will increase the creation and rate of these gates. And so we're just gonna get a bunch of sodium out into the blood. Therefore, water can follow into the blood. And then that's gonna increase blood pressure. Also, ADH, we already talked about it, so I'll just go quick. But remember ADH, it binds to this G protein coupled receptor, act on this aquaporin vesicle, which will bind. So now we have aquaporins. We have an aquaporin in here. Basically just means more water will get out into the blood. So aldosterone reabsorb more salt, therefore more water will come in, and then ADH will increase water as well, creating aquaporins, allowing more water to go into the blood. So that's our ROS system. It's pretty cool. All right, now I'm going to attempt to teach you about urine dilution and concentration and how that works. So if we start here at the glomerulus, we have a concentration of 300 milliosmoles. And that's the same with out here, Well, I'll wait on these. Sheesh. And in this area, we can, so I kind of drew it bad, but just for the sake of teaching this, that's the way I drew it. But we can talk about the PCT and also the PST right around here. Because remember, those don't change concentrations. They stay at 300 in those PCT and PST. But then this loop of Henle right here is a big way we can determine dilution or concentration of the urine. And so we're gonna start with the ascending. So the ascending, remember its job, it has sodium, potassium, and chloride pumps. And so it pumps a bunch of solutes out. And it can pump against a really strong gradient. But it's gonna pump these solutes out and create a concentration of 600 milliosmoles here. And in the tubule, it'll be about 400 and 300 and 200 and 100, as it's impermeable to water and it's pumping a bunch of solutes out. these are the concentrations we're gonna have. But it's gonna set up a gradient in the interstitium. And here it's gonna be 500 milliosmoles, 400 right here, and back to 300, which is our normal up here. So those pumps, those sodium potassium chloride pumps, are gonna establish this gradient everywhere in this interstitium. 400, 500, 600, and out here too. Okay, so we have a gradient established now. So in this descending portion, remember this is permeable to water and we only have aquaporins. So actually, yeah, I'm just gonna, this is in blue. That's a bad blue, that's okay. So this portion is permeable to water. And after the proximal straight tubule, we get into this descending tubule, right? And water is gonna start to leave to match this concentration gradient that's been set up by the ascending tubule. So we were at 300, and water is gonna leave and create a concentration gradient of 400 milliosmoles inside the tubule. So we're at 400, but now we drop, as we drop, we're at now at a 500 outside in the interstitium. It's gonna change to 500. Now we're at 500, it's gonna do the same thing. Water's gonna leave and become 600. So that's how it's set up. And then so we're 600 down here, and then we get up to 100 up here. But now in the collecting duct, hold on here actually. Yeah, we're actually gonna write it out here too. 300, 400, 500, 600. Concentration gradient. So in here in our collecting duct, if you remember, we have aquaporins again. Let's draw that in blue again. And this is how we can dilate or concentrate the urine to be excreted, or to be urinated, to be peed out of the body. So we can keep this very dilute urine and just pee it out, especially if we have a diuretic, right? We're not going to push as many solutes out. We're going to keep this a small concentration and very dilute concentration and pee it out. But if we want to retain that water, our body does the mechanisms that I talked about previously, like the Ross system or the tubuloglomerular feedback, and it's gonna fire ADH. So when we have ADH, remember those vesicles will bind and water basically begins to leave. So yeah, I'll draw it. So we have 100, no, I'm not gonna do that. Terrible marker. So you have 100. But now we want to concentrate the urine, and so we have ADH come and yeah, so so less water goes out. We want water back into the interstitium, back in the blood. So ADH comes and makes those aquaporin vesicles fuse, and so water leaves. Goes back here, so now we're at 300. It's gonna leave, and then we're at 400. And same concept over here, right? It's just gonna match the outside. So we're at 600, and it's very concentrated urine, but we're saving water. We're reabsorbing the water into our blood. And so that's how ADH works on concentrating urine and retaining that water, so we have a high blood volume and high blood pressure. So very cool.